Hello and welcome to this lecture on autochthonous resources. Let me say it again and a little bit slower. Autochthonous resources. Autochthonous resources is a 10th lecture in this series on stream ecology. And here we're going to cover algae primarily, but we'll also talk a bit about macrophytes. As per usual, here are the objectives for this lecture. I want you to understand what autochthonous resources are. You should easily be able to define those by the end of this lecture. I want you to understand how they're important to streams and also when they're important to streams and how they differ from each other. So we're going to talk again about diatoms and plants. I shouldn't say just diatoms, but it's going to very heavily favor diatoms, which is what all these images you keep seeing are. And I want you to understand that there is a real difference in the way that algae, like diatoms, contribute to stream ecology, and plants, like, say, what Valsneria, or what people call eelgrass, also contribute to stream ecology. And then finally, I want you to be able to predict how changes to these resources will impact stream ecology. And that will be obvious and easy to do after you understand how these things contribute to the ecology of the stream itself. All right, so this name is a little burdensome, right? So what does it actually mean? Well, an autochthonous resource is a resource that is found within the region of study. What does that mean? Well, think about it this way. So if I say autochthonous source in a stream, I mean things that are actually produced in the literal stream, resources that are produced in the literal stream itself. What does that mean? Does a tree leaf that's chewed up count? No. A tree leaf is from, and again, this is an artificial line, but we're saying that's coming from a terrestrial system. So the source, the ultimate source of energy for that system is terrestrial in nature. If you compare that for us to what actually grows in a stream, then we have algae that are growing on rocks, and we call that autochthonous. I do want you to start to think about the difficulty because, again, this is a terminology used based on a conceptual model about how systems and energy flows work. And I want you to understand that it's not going to work terribly well in some cases. What do we make of plants that grow at the surface of the stream, like these lily pads here? So they're autochthonous in part is a way to think about them. They are certainly growing in the stream, but they function much more like a terrestrial plant, and their roots are penetrating down into the sediments quite a ways, so that's quite a bit different from most of the other stuff that we're thinking about. And lily pads may be exposed to different levels of water, right? And so they may be closer to the terrestrial system or further away. And if an animal comes and eats some, it may be a terrestrial animal, right? So these systems are far more interlinked than we give them credit for. A tree hanging over the edge of a bank, is that an autochthonous or an allochthonous resource? Its roots are drawing water from the stream. And in drawing water from the stream, they're also drawing nutrients from the stream, probably phosphorus and nitrogen. Their resources are also being dumped directly into the stream when those leaves fall, right? So these lines are, at first glance, very easy to define. As we get in closer and we find a lot of exceptions, it gets harder and harder. But I do want you to be aware that for general purposes, the term allochthonous and this, or I should say autochthonous, and this other term allochthonous that we haven't got to are very common. One of the interesting things about stream systems is that unlike terrestrial systems, I'm not going to focus on plants as much. Obviously, if I'm in a terrestrial system and I want to talk about primary producers and the importance of autochthonous production, then what we need to start to do is talk about plants right away. Right? If I go stand outside and you wave your hand in a direction, almost everything you wave at that is a primary producer is a plant of some kind. There are very few contributors other than plants, and they tend to be very, very limited in the amount that they're actually producing relative to the plants. So the actual driver then of autochthonous contributions is going to be what you see under the water here, this sort of brown covering on the rocks. It's going to be thin layers of algae. Often that golden brown color is a characteristic of diatoms. So the rocks you see in this image, if you look at them above the water column, you'll see that they are sort of gray. And if you look at them below this water, you'll see that they're that sort of greenish gold color. That's very characteristic of diatoms. If you go out to your local stream, you'll see this. If you take your finger and you swipe it across a rock, it'll feel slimy frequently, and you'll often leave a trail, and what you're doing is abrading off the diatoms. Don't worry, they're just algae. There's not a, you don't have to be concerned about that. The diatoms will regrow at some point. So we're going to spend some time especially focused on diatoms, but I'm also going to bring up this other group called the blue-green algae. All right, these are zoomed in images of diatoms. Diatoms can be fairly large. They form really cool shells around their bodies. They are single-celled protists, meaning that they're eukaryotes, meaning that they have a nucleus. So they are very much like us in that way. They're actually more similar to us than they are to bacteria. They account for a considerable amount of the global carbon fixation, as you can see here. 20% is nothing to be sneezed at. That's a huge amount of production. And they have this shell, or what is called a frustule, made out of hydrated silica, which is another sort of fancy way of saying glass with water, right? And these frustules are very strong. I know that you would think, oh, doesn't glass, this small thin sections of glass break? Well, the pressures and the ways that pressures are being applied to these systems are very different than what you're thinking about. It's not like somebody standing on a pane of glass and jumping upside down, right? So these are very, very different systems. But the glass is actually very strong and it protects them, and it actually helps them become resistant to degradation and to consumption. So organisms will frequently eat diatoms, and even some of the diatoms will survive all the way through the gut and come back out the other end and may produce more diatoms, in fact. That brown color is characteristic of diatoms because they are closely related to another group of algae called the brown algae or kelps, right? So these groups are closely related. And so those pigments that those kind of groups use to collect sunlight or energy from sunlight are similar. And so they share some of those characters. Diatoms tend to be a far more golden color. Brown algae, actually, it turns out, or brown algae groups here, I'm going to use this sort of broad grouping because brown algae is their own group, are not terribly closely related to plants, it turns out, although they do photosynthesize, and they do sometimes form structures that look like plants. For instance, kelp look a lot like plants in some ways. But that's convergent evolution. That has nothing to do with the fact that those things are very closely related. Brown algae, in fact, brown algal groups are actually as closely related to plants as we are, almost. And so that gives you some idea of how distantly related they are to one another. So photosynthesis is rare. It has evolved only a couple times. It has evolved in the groups that lead out to these algae over here, which include our diatoms. You'll see there's a number of other things in here. It has evolved in this group over here, right? The red algae and the green algae and then the land plants. And largely other than that, it is fairly rare in and around other places. The reason it's so rare is because photosynthesis is so very difficult to do. And even these, so this is a phylogeny of protists, so this is all nucleated cells, the cells like our own, haven't really truly evolved photosynthesis. They've co-opted photosynthesis. So they've actually taken in an organism that can photosynthesize. Our group, the protists, have not ever evolved a system to fix carbon because it is in fact that difficult to do, right? So the only group that has ever been able to do that is a bacteria. So how did these protists manage to capture these bacteria? If you remember back to POB2, and if you haven't taken POB2, if you remember back to your intro biology, then you may remember this thing called the endosymbiote hypothesis. If you don't remember this at all, what I'd recommend you do is just look it up briefly. But the endosymbiote hypothesis is, at some point, some sort of nucleated cell ate a bacteria that could photosynthesize. And instead of digesting that bacteria, it basically farmed that bacteria in its cell. And that eventually gave rise to this very tight relationship between the cell and this little tiny organelle that we call chloroplast that actually goes about the process of doing photosynthesis. And if you look at chloroplasts, which you can see in plants, they're little green sort of sacs that float around inside the plant. you will find all sorts of features that suggest that this thing is very closely related to a bacteria and not terribly closely related to the plant cell in which it resides, which is very cool. More importantly, some of the other protists weren't satisfied with this single approach, right? So actually, things like brown algae, golden algae are evolved from groups that secondarily ate a protist that had a bacteria in it. So they have characters that show that there's actually a protist. There was a protist at one point, and inside that there is a bacteria that was a bacteria at one point that does the actual photosynthesizing. And all of those things now reside inside of a single protist, like this. So in the original case, a cyanobacteria, which is going to be important for us in our stream ecology stuff, was consumed by some sort of eukaryotic cell, a protist. That chloroplast was not lost, or that bacteria was not lost, and it eventually was farmed to produce the green algae, and the red algae, in fact. At some point, one of the red algae got eaten by another protist, and that red algae was all but lost except that the chloroplast that was inside of the red algae survived, and it gave rise to these other groups. Right? So it's a secondary endosymbiosis. So there's very, very cool stuff going on. So diatoms are really secondary, and they are really the most it's sort of derived version of this, where they have taken in a red algae that had a chloroplast, and the chloroplast was derived from a bacteria that was taken in by the red algae originally. So it's a very long stepwise acquisition of these things. Let's return again to the diatom's actual physical structure. So diatoms have all these things stored away inside their bodies, and they store them in these what are glass discs, or trays really. They look a lot like petri dishes. In fact, some diatoms look almost identical to petri dishes in some ways. I should say they're not one-to-one. But they're little tiny shells that fit together, these frustrules that fit together. One of them has to be larger than the other, otherwise they wouldn't fit in between each other. And that means that every time that the diatom divides, one of those frustrules is going to make another frustule that's slightly smaller than itself. And one of those frustrules will remain the same size. So the original top frustule, this one up here, will always stay the same size. But this one down here is going to make a copy of itself that's slightly smaller. And that's actually going to cause diatoms to need to get rid of their shells occasionally and regrow them because they keep shrinking in size. They have a lot of cool shapes and patterns on them. A lot of people are interested by these dots. Those are actually little tiny holes that allow the cell to poke out of its frustule and stick out little sticky bits that are good at capturing food, that are good at sticking to surfaces, that are good at helping them help them float around in the water column. There's lots of reasons why they have those, but they're very, very pretty. And that's one of the reasons why we saw those pictures earlier. People have been attracted to arranging diatoms on microscope slides and then taking a picture on them because they do produce very, very pretty patterns. So this is what happens when a diatom divides. Diatoms do a lot of asexual division. They produce more and more of themselves. So let's start, where should I start for this? Let's do it up here, normal vegetative division. The diatom divides. One side of that frustule gets smaller and smaller. You can see it gets smaller over here and smaller until they get so small that they eventually lose that frustule, and then they go through the process of a standard sexual reproduction that we think of as normal, but most groups actually do asexual most of the time. And to be fair, most of your body is asexual reproduction. It's just very rarely that sexual reproduction actually occurs. And they exchange, as you can see here, sperm and eggs, and then those will fuse, produce a new cell that will produce that initial cell of that size. So this is a very cool system. They have this weird declining body size issue, and they have bodies made of glass, so all sorts of neat features. This is, in fact, if you have tons and tons and tons of diatoms and you dry them out, you'll get this white powder. Surprise, surprise, it's little tiny pieces of glass. And it should also not surprise you that this white powder is extremely good at abrading things because it's like grinding glass up into a powder. It will cut and abrade away surfaces very easily. So you can get diatomaceous earth and you can use it to scrub surfaces and you'll get a very nice polish on those surfaces. Now, if I mention silicon, right, I've just mentioned a new and critical group, and I've also mentioned a new and critical atomic element for that group. So in your mind, you should be saying to yourself, if there's a critical element for a group, could it be limiting, right? And you might say, well, now that I think about it, no way, because sand, which is silicon dioxide, is so abundant, right? There's no way that silicon is limiting for diatoms. That's ridiculous. Actually, sand is not unlimited in a lot of systems. Okay, so if you go to a lot of streams, you'll find that sand is not terribly abundant. Water also is a universal solvent, and it helps to dissolve even things like silicon dioxide into it. But it should not surprise you that water does not dissolve very much silicon dioxide in it. It's very difficult to do that, in fact. Silicon dioxide dissolves in water very slowly. Hence the reason you can put sand in a fish tank. And it's not like you've set up that fish tank for a few years and you're like, oh, I better replace the sand. All the sand's dissolved, right? It takes literally centuries and centuries and centuries, right? Thousands and thousands and thousands of years after hundreds and hundreds of years to actually get all of that silicon dioxide to dissolve in water. It takes a long, long time. So it turns out silicon dioxide, which is really important for diatoms, can in fact be limiting. There can be places where it is limited, and there can be seasons when it's limited. Diatoms are really, so let me get to that limitation. Diatoms are really good at photosynthesis. Some of the best photosynthesizers out there, and they will outcompete in a stream most other competitors. In fact, if you go out to a stream and somebody says to you, what's growing on the rock? You could probably say diatoms and be mostly right and not even have to look because they are competitively superior to many other groups. And so they will be the predominantly thing growing on the rock. Now you can take them to the lab and actually scrape them off and look at them. And there's lots of cool reasons why you would do that. But I suspect when you do that, you'll find that it's mostly diatoms. All right. Well, that causes a problem. They are part of this thing that we're going to call the periphyton, so the growth of algae on surfaces in the stream. They outcompete everybody else, and when they do that, they start to consume all of the silica that's available, right? They grow really, really fast. They're consuming all the silica. And then at some point they run out of silica and they can't grow anymore, right? They can't make those little glass houses that they live in. And so other groups can then grow, right? So where do we actually see this? If you look at the picture on the right, what I want you to see is in the top panel, you can see nitrogen goes down as the growing season starts. CO2 goes down as the growing season starts. This all makes sense, right? Things are being absorbed. Chlorophyll goes up. You can see there's an upswing here. That makes sense. Lots of plants are, I should say algae are growing in the stream. And in addition to that, if you come down to the bottom panel, look at this black dot here. These are silicon dioxide, and you can see that that substantially declines, right? It declines by more than half. Down here towards the bottom, where it's bottoming out, you can see it gets extremely low. Very, 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 very low. And this is in micromolar, right? So this is really low stuff. Okay. So the amount of silicon dioxide actually available, or here silicon hydroxide, which is silicon OH4, actually available for these diatoms is very, very small at that point. And so they've absorbed it all. So their growth will slow down because it's harder and harder for them to get silicon that they silicate so they can actually grow their shells. Frustrules, sorry. I keep saying shells. I should use the correct language, frustrules. The other thing that's important to know is that silica will be more common near the bottom, right, and less common near the surface. That's because silica is dissolving from sediments, and the sediments are primarily going to have that material around. So it's a fairly simple cycle. It looks a lot like the phosphorus cycle. Rock dissolves into water. Water is either transported down to the ocean and gets lost, or it gets absorbed into an animal. Animals can die and re-release it, or it can be transported out to the ocean. Very, very simple stuff. But that means that as you get out towards the ocean, you're going to see this stuff be really abundant. And when you're in, say, headwater streams, you're going to have it be much more limiting in that case. All right. And in very, very deep streams, and rivers, you may in fact have differences of abundance availability in the water column itself. So the surface may be very limited, and areas down low may have more of it because they will be where that material is interacting with the available material in the system. So far we've talked just about diatoms, and that's fair considering the importance of diatoms to aquatic systems. But there's a bunch of other stuff out there that I've mentioned earlier in this lecture. There's red, browns, and greens algae, and there's also this other group called blue-greens, which are bacteria, so cells without nuclei. The algae, as we generally call them, the red algae, the brown algae, and the green algae are what are called protists, eukaryotes, or cells with nuclei. And we're going to treat multicellular plants separately, and that's going to become obvious as we get there. Very rarely you may run into systems with what I'm calling here multicellular green algae. Now these are cells that are linked together. They're not forming a lot of different structure like a plant would with the different cells, but they do occasionally occur, and these are often green algae, although they can occasionally be other things. One of the things that does form long chains that we do find occasionally in stream systems are blue-green algae, which again are in fact blue-green bacteria, which are cells without nuclei. And if you look at this picture on the right, you will see that inside that cell, there's not much differentiation. It's also important to note that blue-green algae, blue-green bacteria are the first photosynthesizers, the original ones. This is the only group that has ever managed to figure out how to harness sunlight. And as such, it is really important, right? Because its appearance on the surface of the Earth was really critical time for Earth. It produced all of the oxygen you breathe today, and it has helped to support and maintain that oxygen production. They do form long chains of algal cells. These algal cells are all related to each other, right? They're individuals that just divide. And on those chains, there's one type of cell that are different. That's what the arrow is pointing to. Occasionally, they produce these cells, which are called heterocysts, which are different. It literally means different cell, right? So it's notable. We're not very creative in our naming, but there you have it. And inside the heterocyst, they actually also do this other cool process, which is not evolved very often, which is the fixing of nitrogen. So not only can they snatch sunlight and turn it into actual carbon bonds that form energy for them later, but they're able to take this triple bonded nitrogen out of the air, snap it into pieces, and form organic nitrogen out of it. Really interesting stuff. So these species of algae tend not to be nitrogen limited, right? It's costly to fix nitrogen, so that can be a problem. They can take nitrogen out of the water if they want to without paying for it. That's great. But they can fix it when they can't. And so they're basically limited just by phosphorus. If phosphorus is abundant in a system, they can go to town because they don't have the need for silica like diatoms. They don't need nitrogen because they can make their own. And so they're very reliant on a fewer number of things. So beyond all the cool stuff that blue-greens can do, blue-green algae can also produce neurotoxins, which can be harmful to people. You should not eat, play, use, or fish in waters that are highly contaminated with blue-greens. And this is an example of what that looks like when it gets carried away. Okay. Also, I just want to be clear, this person is sampling that green algae. I would recommend wearing a glove if you're sampling blue-green algae that is this dense. Blue-greens also produce a neurotoxin that can be problematic. In fact, for sensitive people, you should wear a mask if you're going to sample it when it's this dense. In fact, if you're all going to sample it when it's this dense, go ahead and send out the undergrads and the graduate students. Let them take the risk. You've earned your place on the shore at this point. No, I'm just kidding. But the sampling of these algae is a little bit complex when they are this abundant because they can be very dangerous to people. And if you let pets go swimming in waters that are this densely packed with those blue-greens, then it can in fact kill them. Things like dogs have regularly died because they've entered streams that are, or lakes and rivers that are this choked with these blue-green algae. One thing you want to be clear about is that these are the algae that are responsible for harmful algal blooms. They occur rarely, as we've mentioned earlier in the course, they occur rarely in streams and rivers, but they do occur, right? Especially where the water starts to slow down and large numbers of these algae can build up. And they are more and more common in areas near large human impacts, urbanized centers, and areas that get a huge amount of agricultural runoff. So they will increasingly become a problem for people. There's a similar phenomenon that occurs in marine and estuary systems. They are also called harmful algal blooms, slightly different species. I do want to point out here that this greenish color does not look very blue. I know. What I will show you on the next picture is that there is in fact a sort of a bluish tint to them, which is why they're called blue-greens. And here's a good example of blue-green algae. So there is in fact at times a bluish tint to them, right? This is not as green fluorescent as you saw in the prior picture. And that blue color is what people call the blue-green element of them. They're common in fish tanks, as you would expect, where phosphorus can be very abundant. Nitrogen may or may not be abundant, and there aren't usually other competitors that are things like diatoms for them to compete with, so they will rapidly cover areas and surfaces. All right, what about all these other groups that I mentioned before? I said, oh, there's green algae and red algae and brown algae. They don't matter, turns out. They're very rare. These systems really don't have them in them to any extreme extent. If you're working in a system with a lot of those members present, not that they don't exist, but if you're working with a lot of those members present, then you can spend time figuring out how they're important to the system. But in general, we just ignore them. There are cool things to learn about them, but for our purposes in this overview where we're doing a stream ecology, right? I'm not talking about a type of stream, not a type of stream ecology, then we are going to move forward and not deal a lot with them. Before I do move on, I just want to remind you from your reading that there are a number of terms that your books refers to for algae. Epiphyte is the one that I hear most. It is algae that grows technically on plants. Epilithian is algae that grows on rocks. You should probably know those two terms. The other ones, all this other stuff, that is really rare stuff. There's some cool things in there like algae that grows on animals. Oh, that's neat. There is plenty of algae that actually does that, but I don't want you to be super concerned about those. So those two terms will be sufficient for you. Important thing about algae to note is that algae rarely live as an individual cell, even though these are single-celled organisms. They often form aggregations. Bacteria and protists can form aggregations. These aggregations are called biofilms. They have special properties that make them resistant to abrasion, to loss, to consumption. They are often very sticky as far as their ability to hold on to something. They're not sticky in the sense like you touch them and you feel like your fingers touching super glue. But what you will find is that if you pass particle by them, the particle will tend to stick in them. So they secrete this matrix that they live inside of. It helps to protect them from grazers because it's hard for the grazers to work through it, right? It takes a lot of energy to move through this matrix. It helps in collecting particulate that they can extract resources from. It helps in preventing them from being washed downstream. And it produces really complex communities. And what I'm basically telling you is the structure and 3D structure that you see in a forest when you look outside, right, and you see these trees going way up into the air, that 3D structure of using lots of area and volume to make a relationship between members that are very different from each other, which is effectively what a forest is, exists in the microscopic world too. You just won't see it when you first look at it. Here's a great picture of that. There's all sorts of things going on under your very nose if you can zoom in and see it. And the very same patterns that you see in things like a forest reappear in this microscopic world. There's predators and there's herbivores and there's things that do something in the middle and there's lots of 3D structure and there's things that crawl and there's things that swim and there's things that hang on other things. And that's very, very neat stuff. And that is usually all forgotten about. So I just want you to be aware if you're interested in biofilms. If you think, wow, biofilms are really cool, they deserve more study, you're absolutely right. They do. And there's a lot of reasons why biofilms are very interesting to humans, not just in streams, but because biofilms also develop in things like IV lines. And that can be really problematic because bacteria that are in IV lines in a biofilm can actually be extremely resistant to things like antibiotics. And they can cause real issues if they get into a patient because it's very hard to treat them, right? And so those are very cool ways in which this 3D structure is important to these different organisms. I just love this picture. This is from Science Magazine. You can see down at the bottom the citation as well. If you get a chance, go ahead and take a look at this up close and see all the cool animals in it, because it's just neat to see how complex and diverse these systems are. And really, we do lose sight of them when we think like, oh, algae, that feeds the bugs feed the fish. What we really should be thinking is, oh, algae, that's a whole world in and of itself, and look at all these cool animals and protists that live in amongst that. All right, so what about the plants? We talked a lot about algae now. What about the plants? Well, plants, of course, what are plants? Plants belong to this group of algae. They're green algae. They're very, very highly advanced green algae. They have a lot of structure. I'm not terribly worried about you knowing the structure of plants. If you've taken an intro biology course, again, you will have seen this over here. I just want to convince you that the structure of plants is far more complex than what you saw with anything else so far. When I showed you a diatom, that was an individual cell. When I showed you long strings of blue-green algae together, there was one single cell that was different in that entire string. But look here, right? In this image, you've got all sorts of different cells doing different things, right? Lots of complexity, and that's just in the stem of the plant. We haven't done the root, we haven't looked at a leaf, we haven't looked at a flower, we haven't looked at a seed, right? So plants, complexity-wise, go far and above everybody else. The important thing that I should stress here now is that plants, well, I'm going to refer to our vascular plants especially, are ones that have these two types of tissue, xylem and phloem. It turns out some plants do not have that, and so those are non-vascular plants. Mosses and bryophytes or liverworts do not have those. Okay, those vascular tissues are effectively the circulatory system of plants. They allow food to go down, right, and be stored in the roots or to be used by other portions of the cell. And they allow water to go up from the roots to the plant body so it can photosynthesize. So if we look at the relatedness of land plants to other things, what you'll find is that there's a bunch of little green things, single cells and some a little bit more complex, that are closely related to it. But plants themselves, down here, are deeply nested, as we might say, within these algae. So they're a very, very highly advanced algal group. And as we've actually already talked about in this course, plants appear on the Earth somewhere about maybe 480 to 430 million years ago, somewhere in that ballpark. Today, land plants are a very, very, very diverse group. They include lots of different species. Most of those, it turns out, tend to be flowering plants on the order of, say, hundreds of thousands or to millions of species, depending on how we divide it up. And there's a lot of other rarer groups out there. But even some of the less diverse groups still have thousands of species in them. They are land plants, right? They are exactly what I'm saying. They are a group of algae that evolved to exploit the land, and then they have since then subsequently secondarily reinvaded water. Unfortunately, it turns out the adaptations that make you good at exploiting the land are not necessarily the adaptations that make you good at exploiting the water. So land plants are not as efficient in some ways at exploiting water as they are at the land surface. We have a number of different groups which have reinvaded the water. It's not like only one group has ever figured out how to get back into the water, but the groups that we often think about are flowering plants because they are some of the more abundant ones out there. If you go out to a stream and you see these, so these are often sort of reddish in color. Don't be confused. These are not a land plant per se. These are just roots, so they're part of a plant. So not a species is actually using it. But let's talk a little about some of the general trends in plants. So advanced plants, so we can think of this like flowering plants. That's what I mean by advanced plants. Generally live in fast flowing waters. Think about what kind of stream water that would generally be in. Primitive groups tend to live in very, very slow flowing waters and often in areas that are only intermittently wet. Or I should say intermittently fully flooded is the way I should describe it. They're almost always wet, but they're only intermittently fully flooded. So you will find things like mosses and liverworts, which are primitive groups, near to headwater streams, even in areas where the stream is only intermittently truly flowing, but the area around it will often remain moist or wet because you're in a forest and there's often canopy cover, and so that stream may dry out. There may be no flowing water, but the ground will stay pretty humid year-round. Plants tend to grow in shallow water, and they tend to be limited by light, especially when they're competing with algae. The plant itself is able to usually outcompete algae. However, algae, of course, can grow on the plant. And so plants often have to invest in compounds to dissuade algae from settling or growing on them, or they have to rely on grazers coming along and feeding on the algae preferentially over the plant. They are usually competitively dominant, so while diatoms are very, very good photosynthesizers, they can't form these large 3D structures, and large plants can shade and grow over them, and so they can be dominant when the water velocities are slow enough to allow large groupings of plants to grow. And one of the major issues that a lot of these plants have is that they are a plant body with a thing like a flower that they are trying to fertilize in water. There's a lot of issues with that. One of the big ones is that seeds aren't great in a unidirectional system, right? Seeds are really better when they can disperse in all directions. If you can only go downstream, that means that you are going to have an issue colonizing upper reaches, right? You're always going to be washing downstream. And this is going to come up to it again when we talk about stream macroinvertebrates and what we call the drift hypothesis, the drift paradox. Okay, it's very easy to go downstream. It turns out it's very difficult to go upstream. And so there's a real issue with that. So plants have this issue with seeds always going downstream. Flowers in an aquatic system are not great. That's not exactly a great design. So there's all sorts of problems. If you rely on pollinators like bees or flies are big pollinators as well, then it turns out that getting them to the center of a water body to pollinate a flower is risky, right? A lot of those animals are going to die. If they're dying frequently, that means that they're not transferring pollen around. That's going to cause issues. If, on the other hand, you're trying to be air-driven or wind-pollinated, and so you're going to use the air to transport material, you're going to have your flowers be sticking out of the water quite a bit, right? And they're also going to have to be in a way such that pollen can land on them. Okay, and that can be difficult in water to make these big structures that sit out of the water and yet are anchored underneath it. So you're going to tend to see those in slower flowing waters. And then on top of that, when you actually go about the process of growing, it's very easy in a stream for the sediment to move around and you can get buried. And many plants probably die when they can't grow back to the surface after getting buried from a lot of events. And it doesn't take much. It can be just a few centimeters of sediment, and that can be enough to prevent a plant from reemerging in, say, the spring when it's died back after the fall. So there really are a lot of issues for plants living in streams. So if we kind of summarize this, what are macrophytes limited by? Well, certainly light, right? So as with any growing ant thing, light is always going to be a problem. Turbidity then is going to be an issue. So turbidity in streams, so the amount of suspended stuff is going to cause issues for plants to grow. The amount of other plants growing above the stream are going to be an issue. So canopy cover is going to be an issue. Plants are also going to be limited by velocity. These macrophytes are going to be very sensitive to being actually physically broken or uprooted by high velocities. And a lot of sections in streams can have high velocities, right? So that's an issue. They're going to be limited by settlement. So in this case, the ability to settle upstream, right? So that's going to be a limitation for them. They may be limited by other macrophytes, so other macrophytes in the system that grow in part or fully submerged. And very rarely plants are limited by pH in systems. So some pHs are completely unsuitable for plants and they can't grow in those systems. Generally, however, macrophytes are not limited by water, and for terrestrial systems, plants are very frequently limited by water availability and when they can grow. They are not limited by CO2. CO2 is very abundant in aquatic systems in the form of inorganic carbon species that you know, right? You know that they form that carboxylic acid, which acts as a buffer in these systems, and you can have a lot of that floating around. It's very easy to convert that back to CO2. Whereas in the air, CO2 is very, very low density, and it requires a lot of effort to trap it. And so there's all sorts of groups of plants that have evolved ways to capture CO2. And in aquatic systems, we just don't see the same kind of pressure to form all sorts of novel systems to capture and hold on to CO2 because it's relatively abundant. They are rarely limited by nutrients. There are some cases where they might be, but we generally don't see that. And one of the most interesting ones are macrophytes do not in streams appear to be very much limited by herbivory. So if you think about going out into a forest, if you spend 10 minutes in that forest, you'll see that there is all sorts of stuff eating trees, plants, and everything else out there, right? Look at it. You just spend a little time turning leaves over. You'll see holes, you'll see cuts, you'll see nibbles, you'll see twigs, right, that have all been chewed on. And if you go into an aquatic system, by and large, herbivory is a relatively rare phenomena for macrophytes, it turns out. Your book does a really nice job then of summarizing some of the things that macrophytes do. And I do want you to really be thoughtful about the reading here because I can't cover all these and I don't want to try. I think it's crazy. But some of the things that macrophytes do in streams then, here they say macrophytes and bryophytes. Bryophytes are non-vascular plants, okay? So when I say macrophytes, I know I mean vascular plants, but usually I just mean plants in general. But aquatic plants often provide shelter, so people will fish near them because they provide habitat for fish, but not necessarily food. They provide a substrate, so something for things to grow on, not least of which is algae, which is in fact eaten by things. They stabilize stream sediments. They reduce and modify velocities in streams. So when water is flowing through plants, it takes more energy. The more energy is transmitted to the plant, right? And so the velocity slows as they move through macrophytes beds, which means that sediment will rain out in macrophyte beds and generally cause areas with lots of macrophytes to get slower and slower with time until they're largely standing, and then possibly the water will divert around them, right? And so this is a way in which macrophyte beds can modify the system, but they themselves are also becoming modified by the system. They are binding lots of nutrients and biomass, and because they're not being eaten, that nutrient is trapped in them for long periods of time, and they're really important as a result of that, especially in, say, the fall when they are dying back, of releasing and causing availability of certain nutrients in the system. Now, again, your book mentions bryophytes, so I think it's worth at least pointing out what these are. Bryophytes are these little things, non-vascular plants. One of the cool systems that you should know about, there's so much to cover in stream ecology, and I constantly feel like I have to give short change to a lot of things, but these things called quaking bogs. Quaking bogs are built from bryophytes, so these plants literally just cover the surface of a stream, and when you go and stand on them, you can walk around, but because they're literally floating on water, they go up and down, so they quake. So if you've ever been on a quaking bog, you can jump up and down. It feels like being on sort of a trampoline with very cruddy springs. It's neat. And when you push it down, you'll see that water starts to infiltrate it. And if you were, say, to cut a core out of it, you would see that there's water just underneath you. You're standing on a plant and not on the sort of solid ground directly. This is sphagnum, which is a type of moss. Sphagnum is converted into this thing called peat, and it can also be converted even further into coal, basically. And it is frequently burned in all those different stages to provide energy. In some areas, there is still historic mining of sphagnum in any of its variety of forms to heat and homes or to power small areas. Sphagnum is going to be really important for climate change. It is really important for climate change because it vastly increases the surface area that plants live on the surface of the water. It's also very productive. It stores a lot of carbon, and there's a lot of carbon stored in it, especially because it keeps dying and settling to the bottom. And so there's a lot of sphagnum bogs that have huge quantities of carbon stored in them. These bogs will tend to disappear as they are penetrated by upper plants that are able to expand into these systems, and that all of that carbon that is stored there may or may not stay there, may be released. So they're going to be really important for climate change reasons. And if we were able to do this, I would take you to a quaking bog. This is definitely borderline stream or a lake, but if you take limnology in the spring, hopefully we'll get a chance to go out and see a quaking bog. They are really neat, and we can walk on them. They're very cool. This is the structure. It's relatively straightforward. It's just a very primitive plant that's stuck together. Basically, it has no roots. It does have something like a stem, and there is some transport of material in it, but it's certainly not the complexity that you see with the higher plants. This is what that actually looks like, that quaking bog. You can see that there's actually a large segment of material that hangs out literally across the stream, and you can walk on that. That's the actual quaking portion of it. All right, so what are we to make of plants? Well, they are really important at times. There's a number of species. Certainly, we don't want to discard them. Often they're important when conditions are good, so when light penetration is able to extend into the water well, and that we get regular flows, right? When you get really erratic flows, then plants are always going to be washed out of the system. So in really flashy systems, highly urbanized systems, you won't generally see them, but in more natural systems, you will see them. They are rarely part of the food web directly, okay, but they do have effects, and those effects are much different than, say, the algae. And they can still enter the aquatic food web through detritus, right? After they die and they start to break down, they're still an important component of the detrital food web. So don't discard them just because they don't immediately get eaten right away. Actually, this helps us a lot as we think about stream food webs. It makes it really simple. It means we basically still only have two sources that everything is relying upon. Algae, and of the algae, it's often diatoms, and tree leaves, because there's a lot of trees around us and they're dumping leaves. And I shouldn't say just trees. There's a lot of plants that are dumping leaves into the systems. Many of them are trees. All right, so it actually makes it easier for us to understand. So you should be very thankful that that is in fact the case. All right, that's the end of autochthonous sources, or I should say the lecture on autochthonous sources. Next up, we will do this thing called allochthonous resources.